Hi everyone, we are going to look at the process of genetic engineering. For this assignment, you will need page 7F of your biology handbook. So we're going to start with an animation of how bacteria can be used to produce human insulin. Human insulin is a hormone that is generally produced in the pancreas of the human body. Pancreas is one of the organs that helps with your digestive system. The types of cells that are used to produce insulin, which helps to regulate your blood sugar, are called beta cells, and they are found in the pancreas. Following along on page 7F, you should be able to answer questions 1 and 2 before we move on. So as you just saw in the animation, for stage 1 of this process of producing insulin using bacteria, the gene for insulin was taken out of the nucleus of a beta cell. So here you are seeing a blue chromosome which came out of the nucleus of one of those pancreatic beta cells, and highlighted in yellow is the specific piece of that chromosome, the gene that we are wanting to use, the insulin gene. Please write that in step 1, stage 1 of your chart. For stage 2 of this process, the insulin gene is cut out from that chromosome using a special type of enzyme called a restriction enzyme. These restriction enzymes look for a specific sequence of nucleotides on the DNA, and when they find that sequence, they cut at a specific location. Write that in for stage 2 on your chart. For stage 3, a bacterial plasmid has been removed from a bacterial cell. You should remember from Module 3 that plasmids are rings of DNA that are found in bacterial cells. As shown in this animation, that plasmid was taken out of the bacteria cell, and it was also cut open using a restriction enzyme. Remember that these restriction enzymes are looking for a specific sequence of DNA, and when that sequence is found, they cut at a specific place. So we cut this donor gene, the insulin gene, with the restriction enzyme. We also cut the plasmid with the restriction enzyme, so they were cut at the same sequence of nucleotides. Write that in for stage 3. For stage 4, those two pieces of DNA, the donor DNA, the insulin gene represented in yellow, and the bacterial plasmid DNA, which is represented in red, were spliced together. They were put together because they were cut at the same sequence. So now they can fit together. That new piece of DNA, that plasmid that has now got a new gene in it, is called a recombinant plasmid. That recombinant plasmid, it has been recombined. It's DNA of the bacteria in red and DNA of the human donor in yellow, has now been put back into the bacterial cell. This bacterial cell is now called a transgenic organism because it contains not only its own DNA in red, but the DNA of another organism, a human, in yellow. It is transgenic, other genic genes, other genes. We can also call this type of organism a genetically modified organism or a GMO. Write that in for stage 4. In this animation, you see that the modified bacterial cell, the transgenic bacteria cell, has now been put into an apparatus called a fermenter. And as shown in the enlargement just a minute ago, that bacteria cell is now reproducing asexually, module 5, asexually, so that it makes identical clones of itself. So if the original bacterial cell has the insulin gene from a human, all of its offspring will also have the insulin gene for a human. So now all of these bacteria cells are producing insulin in large quantities. Insulin is a protein that is a hormone that is needed to help to regulate the blood sugar of organisms. Write that in for stage 5. So now that insulin can be collected, it can be purified, it can be packaged, and it can be used for people who don't make enough insulin for themselves. People who have a disease, for example, called diabetes, and they have to take injections of insulin in order to regulate their blood sugar. In order to produce this insulin so that they can use it, this process is used. It can be producing insulin in very high quantities and in quantities that allow it to be distributed to all of the people who are diabetic who need that drug. Write that in for stage 6.